Hello, my name is Janet Heischetter, and it's my privilege to serve as the Executive Director for the Dystonia Medical Research Foundation and to welcome you to this special program. It's because of your generous support that the DMRF is able to continue to invest in important dystonia research. We're grateful to you for this support and for the confidence you have in the foundation to identify those projects that will get us to our goal of more treatments and ultimately to a cure. Thank you so much for that support, and thank you for making time for being a part of this program. In the 45 years since the DMRF was founded, the organization has invested over $35 million in dystonia research and have supported over 300 separate investigators. The DMRF is proud of the role we have had in critical dystonia discoveries, important collaborations that have been formed among researchers, and in our efforts to engage young investigators to the field of dystonia research. All of this has been made possible because of your support, and we can't thank you enough. This program, we will hear from experts about what is new in dystonia research and what's exciting. And we're really looking forward to this program, and we have a fabulous faculty for this discussion. But before we get started, I have just a couple of announcements. This program is being recorded, and we will send you the link once it's ready and posted. So you can look at it again, or if anything happens to your internet, we've got you covered. The program's going to be recorded. Everyone but our speakers will be muted just because of the number of participants. But we want your questions. Your questions are an important part of this program. So please use the chat or the question function on your screen to submit your questions. And thank you to those of you who have already submitted just really terrific questions. We will get to as many of the questions as we can in the time that we have. So if you have technical difficulties, you can contact my colleague, Jody Roosevelt. Her email is listed here on the screen, and Jody will do her best to make sure that we resolve your problems. But again, the program's being recorded, so not to worry. Okay, so now on to our program. It's my honor to introduce our panel tonight. Dr. Joel Perlmutter is the Elliott Stein Family Professor of Neurology at Washington University. He is also a professor of radiology, physical therapy, and an associate professor of anatomy and neurobiology. Dr. Perlmutter received his BA from Princeton University and his medical degree from the University of Missouri. He did his residency and a neurology fellowship at WashU. In addition to treating patients, Dr. Perlmutter is widely published and has expertise in many areas, including imaging. Dr. Perlmutter is the co-principal investigator of the Dystonia Coalition and has been a longtime member of the DMRF Medical and Scientific Advisory Council and a frequent speaker at patient and community education programs hosted by the foundation. I'm also very proud to say that Dr. Perlmutter serves as the DMRF Scientific Director and is an ex officio member of our board of directors. Next is Dr. Ellen Hess. Dr. Hess is a professor in the departments of pharmacology and neurology at Emory University. She received her BA in psychobiology from Wellesley College and her PhD in neuroscience with a focus on behavioral pharmacology from the University of California in San Diego. Dr. Hess did her postdoctoral work at Scripps Research Institute, where she focused on molecular biology. Her work now focuses on the pathophysiology of movement disorders, utilizing a multidisciplinary approach to determine the contribution of the basal ganglia and the cerebellum to different movement disorders. Dr. Hess is also a member of the DMRF's Medical and Scientific Advisory Council and has been faculty at several of the Foundation's scientific workshops. And to all of our dystonia advocates out there, Dr. Hess is one of the people who has received an award from the Department of Defense Peer Review Medical Research Program that all of you work so hard and continue to work hard to make sure that dystonia is included on a list of eligible conditions for study. So congratulations, Dr. Hess, on that award. Dr. Aasef Shaikh is our next panel member. Dr. Shaikh is Associate Professor, Department of Neurology at Case Western University School of Medicine and is a staff neurologist at University Hospitals in Cleveland. He did a fellowship in movement disorders with a special emphasis on deep brain stimulation at the Cleveland Clinic and was a DMRF clinical fellow in movement disorders at Emory University. Dr. Shaikh also did his residency in neurology and an internship in internal medicine at University Hospital in Cleveland. Dr. Shaikh also serves on the DMRF's Medical and Scientific Advisory Council, only the best for the DMRF's MSAC, and is co-editor-in-chief of the Foundation's new journal, Dystonia, which we'll hear more about tonight. We're very excited about this. He has also been a frequent speaker at DMRF, community education programs and community events. I want to thank all of you for being with us tonight, and also for the work that you do every day to get us closer to our ultimate goal of closing our doors forever. We are so grateful to you for all of your dedicated efforts. At this point, I'll turn the program over to Dr. Perlmutter.
Thank you very much, Janet. It's my pleasure to be here, and I want to thank everybody at the DMRF and all of you who are watching who have helped support us. Without the DMRF, well, this would be a far different environment for dystonia right now, and I want to review that. But just let me also shout out not only to Janet, but to Jody and all of her work getting this together, as well as Jessica. So there's a wide group of people at the DMRF who have made this possible. So how has the DMRF really helped with dystonia research? I would say very clearly, without the DMRF, we would probably be very little on the path to finding new treatments. We would not have developed many of the treatments we have. And so what I'd like to focus on is the research that DMRF has provided and promoted, the advocacy that's been important. I'm going to explain exactly what I mean by that, the training of people, and by that I mean investigators and clinicians, as well as the improvement for education for patients and families. So all of these have been important components of the mission of the DMRF. So let's just talk about research first and some of the exciting research. So Janet told us 45 years, millions of dollars forward, and that has made a huge difference. So we all know about genetics of dystonia that were first discovered with the support of the DMRF, and a number of different genes that have been discovered. And now trying to look at, for example, how DYT1, that causes a very common childhood onset dystonia, how that is affecting specific types of nerve cells in the brain. This is the focus of a new grant just given to Zandra Brakefield at Massachusetts General Hospital, part of the Harvard system. This is now 20 years or more after the first discovery of DYT1, that first gene known to cause dystonia, but we're still investigating how does that lead to changes in the brain that subsequently cause manifestations of dystonia, and more importantly, how can we target specific parts of that pathway to help bring new types of treatments. And so it's this basic research going back to Zandra Brakefield, whose lab that Laurie Ozelius was initially in when they first found the DYT1 gene. So the support goes to new people and new investigators, as well as to established investigators when they have new ideas. That's the kind of stuff that's being supported by the DMRF. And in fact, that takes us to just a recent Dystonia Coalition Career Development Award. And so here is finding a new young investigator. And this one is Nareen Bukhari Parla-Turk. I hope I said that right. And she's at Duke University, again, helping to establish a new investigator. This kind of support not only helps the specific research projects that she's doing, but it brings on board a new person who may get connected with dystonia research. So it's the gift that keeps giving. That person may continue with a career's worth of research on dystonia and training other people. So there's a multiplier effect that's incredibly important. And so that really brings us to the training. But advocacy is so important because what we've seen from the DMRF is money directly to individual grants and to training. But perhaps what has even a greater influence is advocacy. And that advocacy we've now seen, and we just heard, going to the Department of Defense, convincing them that dystonia is one of those conditions that they should promote research dollars for. And so then we've seen Ellen Hess get that, and a number of different investigators now being funded by that. And I can tell you my personal story. My first NIH grant that I wrote for dystonia back in the 80s, I should maybe not say that, back in the 80s, it was reviewed at NIH by the peer review system, was thought to be really good science, wasn't even close to being funded because the main criticism, and I quote, dystonia is not an important condition. That was the only criticism. DMRF went to bat. They started advocating. I just resubmitted the grant. Son of a gun if it didn't get funded at one of the highest levels. So it was the kind of advocacy makes a difference in not only Department of Defense, but National Institutes of Health, which turns into large grants like the Dystonia Coalition. And we'll come to that in a moment. Patient education, again, incredibly important. And hopefully you're all receiving that as we go through this, and we'll have questions specifically on the kinds of things that you're interested in. Other new exciting developments, we're going to hear from Ellen Hess about the role of the cerebellum. So when I was starting out, the main issue, focus in the brain, was a part of the brain we call the basal ganglia. And now we're learning more how the cerebellum may play an important role also in the development of dystonia. And then looking at very specific issues in those pathways. And that's really a very exciting new area of work being done. So new grants, new research, new investigators, advocacy that continues to go. And now we also have a new journal. And so I'm going to turn it over to Aasef Shaikh, who's going to tell us about the new journal, which is an incredibly exciting thing, because we've had to send articles about dystonia in a variety of different journals. There was no one home for our dystonia research, and that's where we can go. And so I would like to then lead into let Aasef take it away.
Thank you, Dr. Perlmutter. Thank you all for joining us today. It's such an honor and a pleasure to be here discussing about something that was not just my dream, but many of our dreams who do the work in the field of dystonia, to have a dedicated journal on dystonia. The problem with dystonia is that it is so far common. It is about 3 million people suffer from dystonia, and it is one of the third most common movement disorders after tremor and Parkinson. And although it is extremely common, there was no specific journal which was dedicated to dystonia. I went on PubMed and I did a little analysis of how many research papers were published on a common journal, a common condition, tremor. And then I went in to ask the same question: how many research papers were published on a condition, dystonia? And I plotted a little graph, and there was an exponential increase. And believe me or not, number of papers each year on dystonia was very similar to number of papers that were published on tremor. We had dedicated journal on tremor, we have dedicated journal on Parkinson, but we did not have a dedicated journal on dystonia. And this was one of the biggest motivation for us to come up with the journal Dystonia. The another beauty of dystonia is that the field has a prominent scientists, basic scientists like Ellen Hess and many of our colleagues who are PhDs. Many of them are geneticists, many of them do molecular biology, animal models, single unit physiology, mathematical modeling. And on the other hand, there are a lot of clinicians who do research on botulinum toxin, how DBS works. So the field is quite vast, from a molecule, a cell, a petri dish, to a human and a drug trial. So we need a common home for that can accommodate everything. So if the biggest challenge that happens to us as a translational scientist, that we go and we try to publish our work in clinical world, and often we are told that, oh, this is too crazy idea. And then when we try to publish that in a basic science, then they will say, oh, we know this already. So we need something that takes more translational work. And that was a third challenge. So then we thought that why not to come up with a journal, Dystonia. The other challenge that comes across is when we try to publish, a lot of journals look at the page count. We are not allowed to publish more than 2,000 words. And if I want to put together a detailed science, 2,000 words is just my method. So we wanted a journal which can accommodate comprehensive papers, which can give transparent science to the public. And best of all, we wanted to make it accessible to anybody, not just someone who is affiliated with the university who can just access the journal to the library. So then we came up with a very, very exciting model of open access model through Frontiers, which is one of the major publisher in not only in science, but many other subspecialty they publish. But the good thing about Frontiers, it's open access. That means anybody wants to, let's say Dr. Hess published a paper on animal model of an interesting form of dystonia, and somebody from Cambodia wants to look at it, they can just simply go online and download that paper. Not that Cambodia does not have library. I'm just saying anybody in anywhere in the world will have access to paper for free. And that's the best way to practice science. And it's not only free, but it will also have every detail possible that we want to tell our world to reproduce the experiment and to understand it well. So that was one of our dreams. The other problem that we come across is when we try to publish in open access, somebody has to pay the costs, right? Because those are free papers, so nobody can really charge money for it. So then open access papers often are very expensive to publish. They charge in the range of $1,500, $2,000, $3,000, and many people cannot afford to publish in that. Not everybody are privileged with a research funding to publish that. So we had other arrangement with DMRF that people who cannot afford to publish open access, which comes with a fee of about $1,500, DMRF was gracious enough to cover those fees for those who cannot afford it. So this was like a dream come true. Like you can have a paper with freedom of publishing really good science and not worrying about the cost of publishing it and making your gold standard word accessible to everyone. Now, I'm a translational scientist. I work with neurophysiology and humans and deep brain stimulation and some human physiology. I am not the expert in animal models. I'm not the expert in the basic sciences. So I had to find a partner. And I found an amazing partner, Roy Silito, who is also a DMRF scientific advisory board member, but his area of expertise is basic science. So now we have two editor-in-chiefs. One is basic science and one is clinical science, and we both do somewhat in between translational. And that is going to be a very good model, I believe, to run this journal, which has quite vast scope. And not to mention, we have advisory panel, Dr. Perlmutter, Dr. Hess. A lot of our colleagues are helping us to advising us with their wisdom about certain strategic questions that we would come across to publish a journal. We have a team of 6 editors who will be helping us to handle each paper, because you can imagine it's a lot of work for 2 people to manage with field being so wide. And 6 editors that we chose, they are handling editors. They are experts in a given field. And so every paper, our goal is to give every paper a very fair justice. And that was the whole idea, to put together the team of multiple handling editors who know exactly what's going on, that subspecialty of a subspecialty dystonia. So we are very much looking forward to this journal, and we are very excited about it. We are putting together the first table of content as we speak, and we are hoping to see that and share with you all in a very near future. And I want to thank you all for your support. Without it, nothing is possible. Thank you.
And now we'll hear from Ellen Hess.
Yes.
Well, good evening, everybody. Thanks so much for the invitation to speak. It's quite an honor to be here. And I appreciate you taking the time out of your evening to hear from us. So I'm a basic scientist, as you've already gathered, and I'm going to talk a little bit about drug discovery and where the research is with drug discovery in dystonia. So I think first we need to really talk about how drugs are discovered, right? So there are a lot of different approaches, and the one that you all may be most familiar with is the trial and error approach, right? That's when your physician says, well, that drug didn't work, why don't we try this one? And I'm guessing you all also know that that trial and error approach sometimes works, but usually doesn't, and doesn't work so well. So one of the alternative approaches to finding a drug that might be effective for dystonia in particular is to start with a drug that seems to work, but maybe it's not as effective as we might like, or maybe it has side effects that just don't work for everybody, right? So today I'm going to focus on one of those drugs that we know is effective, which is Artane. I'm going to use Artane as an example of our efforts to take it and improve on that existing treatment. So why Artane, right? Well, it's one of few orally available drugs, that is a drug you can take with a pill as opposed to Botox, that can be effective for treating dystonia. And in fact, Artane is the only drug that's proven effective for dystonia in a double-blind, placebo-controlled clinical trial. So what's that? So a double-blind, placebo-controlled trial is a test of a drug where the patients don't know, or the participants don't know if they're receiving the drug or something that looks like a drug but doesn't contain any active substance. And importantly, the people running the trial, the researchers, don't know who's getting the actual drug and who's getting the placebo. So that's why it's double blind. And what's important about double blinding is neither the researcher nor the participants are biased, thinking, "Ooh, I got the drug," or the participant got the drug, so it must be working, right? So it takes the bias out of it, and that is considered kind of the gold standard to determine if a drug is effective. And Artane is the one drug that's been proven effective with this gold standard trial. So that's why kind of the field of pharmacologists is using Artane as a starting point to develop new drugs, better drugs for treating dystonia. So I think the first thing we have to admit is that Artane is a flawed drug, right? While it may be effective for many people, it has intolerable side effects, right? Constipation, memory problems, dry mouth, urinary retention, the list goes on. And those side effects are so bad, many of you know you've taken it, that they are treatment limiting. And what that means is that most people stop taking Artane because of the side effects. And that's pretty much as good as having no drug at all, right? So the question is, can we develop a drug like Artane that might be effective in alleviating dystonia, but without the side effects, right? And that's the trick. So now I have to get a little sciencey on you guys to kind of give you some idea of how we approach that problem as scientists and where the research is going. Okay, Joel, at this point, I'm going to call on you to call me out if I get too science nerdy, but hopefully I won't geek out on you guys. So how does Artane work? So Artane is an anticholinergic, and what that means is it blocks a signal being sent by the neurotransmitter acetylcholine. So acetylcholine gets released from a nerve cell, and that once that acetylcholine gets released, it tries to find its receptors on a receiving cell, right? And it's only when it actually binds those receptors on that receiving cell, that the message is complete and transferred to the next neuron down the line, right? So what Artane does is it blocks that signal. It actually prevents acetylcholine from interacting with the receptors, so the signal just can't get through. And that's how it does the job of alleviating dystonia. The thing is that there are at least 5 different flavors, different types of acetylcholine receptors that are found throughout the brain and also throughout the body. And scientists have cleverly labeled them 1, 2, 3, 4, and 5. And we know that some of those receptors are involved in the anti-dystonic effects of Artane, whereas other of those acetylcholine receptors are involved in the side effects of Artane, right? So the goal then is to develop drugs that act at the receptors important for the anti-dystonic effects, but not at the receptors that cause the side effects. And we're pretty sure that we know which flavors of those receptors are involved in each of those things. So we think that receptor 1 and 4 are probably the receptors we want to be targeting for those anti-dystonic effects. And we know that receptors 2 and 3 are the ones that are really a big problem for the side effects. So we're staying away from those receptors as we try and develop new drugs. The exciting news is that in the past few years, both drug companies and academic researchers have had quite a bit of success in achieving that goal. So the way it works is that medicinal chemists create hundreds and thousands of new molecules, new compounds that look like drugs, and there are very kind of druggable structures that they're going for. So there are these hundreds of thousands of compounds, and then they're all screened through to see what their effects are on these various cholinergic receptors. Probably there are more than a handful of potential drugs out there now that seem to have the effects that we want. So we have been honored in my lab to have tested many of these compounds, these new compounds, in our animal models of dystonia, and we've actually identified several that alleviate the dystonia in mice. And so that's one step of many in the process of FDA approval, where a new drug has to be demonstrated effective in animals before it goes into people. So there are huge teams of people involved in this type of drug discovery, from the medicinal chemists making those new molecules to the pharmacologists testing them to see which is the one that we really want to, go forward with, and which ones we want to discard and not use anymore. Of course, these teams of researchers span across the US and across the world. A lot of this work has been facilitated by the DMRF, playing a key role not only in directly funding grants to find these new compounds, but also, really importantly, and as you heard Dr. Perlmutter mention already, their advocacy, DMRF advocacy at Department of Defense, which has awarded large grants to promote this drug discovery effort, and at the NIH also. Not only am I a recipient of actually 2 DoD awards for our work on dystonia, which has helped so much, but for example, Dr. Jeff Kahn's group at Vanderbilt is one of those groups that's actually working to make new molecules, new drugs that can be used, that we can then test in our animal models. So with that, I will stop and pass the baton back to Dr. Perlmutter.
Okay, thank you very much, Alan. Thank you very much, Aasef. And I think we're open for questions. And I'm sorry, I only knew about one of the DoD grants and didn't recall about the other, but I think that's great. But I was looking for epaulets on your sides and see, should I call you Colonel or salute? But you know what?
It's general.
Support
Joel.
from all sorts, from all sources is very valuable and helpful.
Yeah, and actually, I would say, Dr. Perlmutter, we know that there actually over $17 million has been awarded through the Department of Defense program to dystonia investigators. So Dr. Hess was one of them. Dr. Khan was another. So Dr. Peterson, some people may have heard his webinar earlier this year. So advocacy works, and we're really, really excited about that. So I have a question for the panel to kick us off here. Overall, what are you most excited about right now in dystonia research? Dr. Perlmutter, you want to start?
Okay, I guess I'm going to start. So there are lots of exciting things, and so that's a difficult question to answer. But I'm going to give you a couple of different things. So I'm hugely biased. I think one of the biggest, most exciting things right now is the Dystonia Coalition. And the reason I'm so excited about that is it's bringing together investigators from all around the world who at times would work together, at times work competitively, and bringing them together to work collaboratively and bringing together the DMRF and other patient advocacy groups, all for the mission of helping people with dystonia and pushing this research forward. That's one thing. But the other thing, which is there's so many cool things going on, the stuff that Ellen was talking to us about, that Jeff Kahn's working on, trying to sort out the very specific kinds of cholinergic receptors to separate out the benefit from the side effects. Listen, I've been giving trihexyphenidyl, which is the generic name for Artane, by the way. That's what I always use. And for many, many years, and it would take at least a year to build up to the drug dose that would be tolerated by a person because of the side effects. And now we have an opportunity to really go at it, looking at, I could go on, brain networks now is a whole new way of looking at brain networks. And it's just, that's going to help us target new approaches for treatment. But I'm going to stop there and I'm going to pass the baton. Ellen?
Yeah, so what I didn't mention is that I'm really excited because some of these compounds that I was talking about, these new drugs, are actually, clinical trials are being planned now to test the safety of those drugs. So we hope to see these drugs into people in the next couple years, and then actually test for efficacy. That is, do they work to help alleviate dystonia? Probably a few years down the line, but we are closer than we have ever been in terms of getting some new orally available drugs that can be taken in pill form to patients. So that's incredibly exciting and rewarding to be part of that research.
That's great. Dr. Shaikh? Yeah.
Of course, the first most exciting thing in my world right now is Dystonia Journal, not to mention. But from the scientific standpoint, I think machine learning and artificial intelligence, that is also, we had a paper which came from Dystonia Coalition, about 2,300 patients with dystonia, world's largest probably cohort so far. We did machine learning, artificial intelligence analysis. It does miracles. It gives a lot of unanswered questions are answered with that paper. And us and many other groups are also interested in using AI to separate various forms of tremor, various forms of phenotypes of dystonia from the physiology data. And last but not least, we also use machine learning to separate the cellular characteristics of the cells in the brain in patients with dystonia, to separate which cells are problematic versus which are not problematic, and how we can separate different clusters of neurons in dystonic patients, versus non-dystonic patients. So that is another tool that we have available, which is very hot nowadays. And that is something that we and many others are applying, which is also very exciting. I wanted to make one correction. I did say 3 million people with dystonia. It's actually 300,000. I apologize. On the note of advocacy, it is wonderful that we have Department of Defense convinced that they need to fund dystonia grant. One disappointment that I have is Department of Veterans Affairs, which is when these people retire. Unfortunately, it's not seen that favorably, and we really need to work in that direction to convince the Department of Veterans Affairs. I work at VA, and I once looked up on the VA database that how many dystonia grant was funded in last 20 years. My answer was 1. So it is something we really need to work on too, because it is extremely common in veterans. And I see that firsthand because I run Movement Disorder Center in the major VA hospital.
Right. And I think you raise a really important point. And again, to our advocates who are joining us tonight, this is on our agenda for the year. COVID has gotten in the way a little bit, but it's something that the Dystonia Advocacy Network and the organizations that advocate on behalf of the community are working on. So we hope in a year's time we'll have good news for you on that score, Dr. Shaikh. So thank you for that. But I have another question for you. Mm-hmm. What is the status of the use of transcranial magnetic stimulation, TMS, to treat dystonia and movement disorders? And then can it also be used for someone who has dystonia and depression? Where are we with that?
So I will answer the easy question first. For depression, we know that it is already in clinical practice. So our psychiatry colleagues use this TMS to treat depression. Now, TMS and dystonia, that is a difficult question. And I'm saying it is a difficult question because there are numerous studies, numerous grants often we review, numerous papers we review. They all have different hypotheses how TMS can work and how it cannot work. We still don't have a very clear answer whether we can really use TMS to treat dystonia the way we use it to treat depression. I must say that we can definitely, and there are several studies out there which uses TMS as a tool to study the brain activity in dystonia. But I would be cautious to say that we are ready to treat dystonia with DBS. And with that note, there are some other studies, including our studies, which we are looking at modulating proprioception with vibration. So, and after that, a few other studies came out. Again, like we need much more work before these non-invasive modalities would get to the clinic, but we are very hopeful for those.
Right, so there's promise and we just need more work there. Yeah, that's good to know. It's good to know. Dr. Perlmutter, we've had a couple of questions about the spread of dystonia. So whether it started as cervical dystonia and has spread to the oral mandibular or to laryngeal dystonia or focal hand dystonia, what does our research tell us about spread?
So that's exactly where the Dystonia Coalition has become incredibly valuable. Up to this point, all of our data and information about that was what we call retrospective data, or looking back. People would come in and tell us what had happened to them 15 years ago, 20 years ago. So it was based upon their recollection. And now we're collecting prospective data where we see somebody and then follow them forward and see what kinds of spread there is in people. So looking at progression of dystonia, one easier method of determining progression is whether they're spread to different parts of the body. And that's what the coalition has shown. And what we found in that, in our studies of that, which confirm some of the retrospective type studies, is that people who have dystonia affecting particularly around the eyelids or blepharospasm, they're more likely to spread to lower part of the face and to the neck than people, for example, who start in a hand or in the voice box with laryngeal dystonia. They still can spread, but not as commonly as those with starting in the eyelid. So it's different. The risk of spread is different depending upon where that dystonia begins, as well as what age it begins. And so that's another big factor. So we're learning more about that. And most of the data, which we used to say, if it's going to spread, it's going to happen in the first 3 to 5 years. But that was biased data because that's what people would recall. And now we're finding out a little bit more about spread that may be delayed, but it still seems to be much less common. So we're learning a lot about that. And that's really important if we want to develop a treatment that helps to prevent spread, because we have to know how often it occurs to see if we're actually making a difference in people.
So can you say where it starts and at the age of onset? Are those predictive factors? Are we at a position where we could actually say that now? It'll predict spread?
So we can say that based upon what part of the body it begins with, we can say what the risk is of spread over time, given the data that we currently have. So we're not bad at it, but we don't have enough data to say to an individual, you will spread or you will not spread.
Okay. Good to know. Yeah. What do we know about whether or not messenger RNA, mRNA is a good candidate for a treatment for a genetic dystonia? Do we know anything about whether or not that's applicable too early?
Well, there's a couple. I can take this or somebody else, but there's a couple of things here. First of all, my guess, and this could be a wrong guess, is the mRNA question stems from mRNA vaccines that are the hot thing today. And so this is really different. We're not talking about vaccinating against dystonia. That would be potentially relevant if there was spread of an abnormal protein. One could vaccinate against that kind of protein. It's a little on the craziest side, but a possibility. And some disorders are going at it, but we don't know enough about dystonia to do that. Particularly, most dystonia is not genetically identified causes, and so that wouldn't be relevant. On the other hand, then there's a whole different category of using genetic type treatments. And so one is using or trying to hit mRNA, and that is in people who have, for example, Huntington's disease. And Huntington's disease, we know, is caused by a specific genetic abnormality in the DNA. And the DNA then codes for an abnormal mRNA, and the mRNA then codes for the abnormal protein. And so what's been in trials recently, although just recently stopped, I might add, for a variety of reasons we'll talk about, is if we inject into a person's spinal fluid an mRNA type drug that can go to the abnormal mRNA in that person's body and block it from then producing abnormal protein, that has a rationale for trying to treat the condition. And what's been found in early studies in Huntington's, one can do that give different amounts of that mRNA. One can see a different amount of the abnormal Huntington protein from being produced, as if it's going to help. Now, then there was a big phase 3 or a much larger trial looking at that, which was then stopped early on, not, we're told, for a safety factor, but we haven't been told exactly how come, although we suspect it didn't provide the benefit that they were hoping to see. And there could be a lot of reasons for that. They were targeting mostly all kinds of mRNA, whether it was the abnormal or the good mRNA that makes actually normal Huntington rather than abnormal Huntington. So there are different kinds of issues, but mRNA treatment is the kind of thing that would be relevant usually for something that's caused by a very specific genetic defect. And we don't have very many specific genetic defects that affect a lot of people with dystonia. DYT1 is such an abnormality, but we already know that 2/3 of those people never get the dystonia anyway. So we need to figure out much more about the condition before we go into that direction, I think.
Right, and along those lines, we have a question from someone who is a twin and their twin has dystonia and they do not, and just wanting to know how they can together, help to advance our understanding of dystonia. So what advice do we give to people generally who really want to get involved in research but don't know where to go or how to do that?
Well, let me give you several possibilities. First of all, if an individual wants to know what research is going on in dystonia, there's a website, clinicaltrials.gov. And anybody can go to that website and see what research is going on. The caveat here is those are not necessarily studies that are approved by NIH or any federal agency. They're just registered. So then you need to identify something on that trial site and then talk to your physician about that to see if it's a reasonable trial, because I can tell you I've seen plenty of crazy trials there that make no sense. So that's really how an individual can do it. Now, coming back to the question about twins, if they're identical twins or non-identical twins, it makes a difference. Mm-hmm. But there are a number of issues going on. So first thing that's really amazing, and I think there were some of the other questions that touched on this, is if somebody has the abnormal mutation for DYT1 or TOR1A, if we call it that now, and they have that abnormal gene, they only have about a 30 to 35% chance of ever developing manifestations. How come? So there people are looking now at what the molecular factors in the brain are that help to determine whether somebody will or won't develop that. If we had a bunch of identical twins, one with and one with, both with the abnormal gene, if they're identical, then try to identify what's different genetically or epigenetically or with other factors going on, that could help sort this out. Because if we could find out the factor, maybe we could use that factor to prevent everybody from developing manifestations. So there is an idea for doing that research. There is potential for those people participating, but what we need is a study that can acquire enough people to make that kind of research viable. And that's not easy. So it's hard to write a grant to say, I'm going to find only identical twins with DYT1 in both, because nobody's going to believe you can get enough people to do that. But the registry that's been done by the patient advocacy groups, if we have a registry and we could find in a registry, all of a sudden there's 100 twin pairs, you'll get a grant written by some investigator to take advantage of that and reach out to those people to participate.
Sure, and I'll just give a plug for the registry. Dr. Perlmutter is referencing the Global Dystonia Registry. That is a collaborative effort of all dystonia patient advocacy groups coming together, and we can send everybody the information, the link to it. You can enroll, unenroll at any time, but it really is helpful for us finding these kinds of things. So, Dr. Hess, we have a question about how do you give a mouse dystonia? I mean, papers have been written on this. So could you just briefly sort of give us the lay version of how can you give a mouse dystonia?
Right. Yeah. So this is how much time do you got? Right. So this is a career's worth of question actually being asked. So the straightforward answer is that since the geneticists have done such a good job of identifying genes that cause dystonia in people, we then actually have the ability to put those very same genes, disease-causing genes, into mice. And if we're really lucky, the mice have dystonia also. And so we do have models like that. Other models, like Dr. Shaikh was saying, we kind of know which parts of the brain are involved in dystonia, right? So we can disrupt the signaling in those specific parts of the brain in mice and induce dystonia that way. We can also just give drugs not experimental drugs that cause dystonia. So we have multiple ways of doing that, and it depends kind of on what question we're asking? Are we looking for a treatment? Are we looking for how that part of the brain is misbehaving? What's our question? That is kind of what drives our choice of models. So we have lots of different options.
Yeah, thank you.
I want to add one thing. Dr.
Yeah, please.
Gina and I once induced dystonia in normal people, normal humans, by vibrating their necks. That can happen too.
So that to your earlier point about that whole new field of study with
Yeah,
vibration
so-
too. Yeah.
Yeah, if vibration can cause dystonia, now we can use vibration to treat dystonia.
Right. We have never tried vibration in tiny mouse necks.
No, probably not a good probably not a good idea, and not something that would be welcomed by most people either, Dr. Shaikh. So we don't want you to cause dystonia. We want you to work on getting rid of it. So, but I have a question for you, Dr. Shaikh. What do we know about research that's being conducted to find a link or a relationship between hormones and dystonia? We hear anecdotally from people all the time, especially in women, that hormones have triggered dystonic symptoms. Do we know anything about that?
To be honest, I don't know much about that. There can be many reasons for that to trigger dystonias. I mean, hormone can change a lot of neural circuits, neural firing behavior, and we know that most of neural circuits are connected. Dr. Hess?
So you're anxious to get out of this one, huh? Passing it off so quickly. Wow. Yeah, so I can actually comment on this because as you all know, dystonia is more common in women than in men, and it has been kind of an overlooked subject of research for a long time. We actually decided to tackle this problem not long ago. And what we found in our mouse models, again, is that, for example, estrogen will make dystonia worse in some of those models. And what is known is that in the basal ganglia, that part of the brain that controls movement, actually has estrogen receptors on those cells. And those estrogen receptors change the firing properties of the cells in that part of the brain. So that's like almost a direct line, right, from estrogen effects, ovarian hormone effects, to firing rates in the brain, to dystonia. And so what we did was we dug a little deeper and we asked, well, how are these cells different on a molecular level, from males from females? And actually what we just found, this is kind of hot off the presses, is that the basal ganglia, the part that controls movement in males and females, are actually quite different on the cellular level. And we can kind of change those differences with ovarian hormones. So stay tuned for more, but there's kind of every reason to believe that ovarian hormones have a very profound effect on how the brain responds. And every reason to believe that we actually have enough data right now in various different fields to pull together kind of a credible story about how those hormones are affecting the brain. The problem is it has not been a focus of NIH researchers, DoD researchers, academic researchers in general have not paid attention to sex differences in dystonia, although we know they're there. And the NIH actually is promoting this now. So hopefully we'll have even more answers in the coming not too distant future, actually in the next few years.
Yeah, that's great. That's really helpful. Thank you. Dr. Perlmutter, question for you. An interesting question of emerging possible connections between the gut biome and some neurological disorders. A lot of talk about this with Parkinson's disease. Is there any thought or any investigation going on now as it relates to that and its relationship to possible dystonias?
So there are a number of issues with regard to the microbiome, and that refers to the bacterial contents of the GI tract. And that has impact on a number of chemicals that enter into the bloodstream and then subsequently get into the brain, or chemicals that are actually made in the gut and then get into the bloodstream. So there's substantial differences. And now the machine learning and artificial intelligence type activity to analyze the genetics of these bacteria permit us to do these kinds of studies, which are ongoing in Parkinson's disease. So far, I've told you zero about dystonia, haven't I?
Yeah.
It's very clever of me. So the people have actually, I've actually had 2 people at WashU ask me about this and been interested in trying to look at a study. So this is a challenge because we need to have to get a study like that funded, you generally try to have a focused question or hypothesis. And in this particular case, I haven't had anybody come up with a really good hypothesis or any hypothesis other than, well, we have this method, let's apply it. Now, it turns out that, and sometimes reviewers of grants will call that a fishing expedition, which is not a good thing to have your grant called. It makes it unlikely to be funded. Now, I would just point out that the Human Genome Project, which has been incredibly miraculous, was a fishing expedition. There wasn't specific hypotheses other than, well, let's build it and they will come. And it turned out an incredibly valuable resource. So these kinds of studies are increasingly easy to do. One just has to collect all the poop that is needed for this from the particular participants. And to do the analyses is pretty standard. So this kind of research will start moving forward in dystonia. I'm not aware of anybody doing it right now, pardon the pun, but this may have promise.
And I think it speaks to being open to any avenue of research, right? Which I think is part of what the DMRF MSAC has really tried to do, is to be open and allow for innovation and exploration.
So for example, if identical twins both had the DYT1 gene abnormality, maybe it's their gut microbiome that prevents one from developing manifestations or leads to somebody causing it. That would be a perfectly reasonable hypothesis to test if one had a bunch of genetic identical twins to do that.
Yeah.
But that would be a very reasonable place to look.
Right. I want to be mindful of time here, but we have gotten a couple of questions tonight, and we get these questions fairly regularly at the office about what we know about COVID vaccinations and dystonia triggered symptoms. So Dr. Shaikh, Dr. Perlmutter, would one of you like to comment on that? Because I think it's important. It's very topical right now.
We can both comment a bit here and there, but often my patients with movement disorders ask me this question, whether they should take COVID vaccine or not. And my personal preference and my answer to them is go for it, because yes, when you take, when I took COVID vaccine myself, I was worn out for the whole day. Yes, you will have some transient worsening of dystonic symptoms just because you are wearing off, because you're not feeling well because you had vaccine, but it's a temporary thing. And I personally feel that it's more important. I have not come across any obvious papers which has shown any direct relationship between COVID vaccine and dystonias or any movement disorder in general, but I think it is important for our patients to take it.
But let me be more direct. Everybody should get vaccinated. And the risk of dying from COVID or having long-term symptoms are substantial. Having a day or two of a little bit more severe symptoms is probably less likely the more mature one is. So for those with dystonia that are my age, the risk of symptoms from the vaccine are very small. And so get the vaccine. That's how our country will get back to operating and the economy going, and you're way safer. We've never had a safer vaccine than these mRNA vaccines. It's absolutely incredible. This technology is amazing technology. This will save lives.
Thank you for that. Thank you for the questions. I think they're important and, as I said, really topical. So I'd like to, we had one comment from somebody who said, I want to thank the doctors for their help in answering some of the questions we all have at this critical time. I do enjoy these webinar sessions. Thank you again for all that had questions and the wonderful doctors that participated. I could not have said it better. Let me just echo this person's sentiment. Thank you to everyone for your support of the foundation's work. Thank you for tuning in tonight. Thank you to our wonderful, wonderful presenters. We are grateful to you again for being with us for this program and for the work that you do. We're going to get there, and we appreciate all of your efforts. So thank you very much, everyone. Have a nice evening. Bye-bye.
Bye-bye.
Bye.